The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Now, before we get started, I'd like to introduce our speaker. Dr. David Beversdorf is currently involved in research on autism, dementia, cognitive effects of stress, the cognitive neuroscience of problem-solving ability, functional neuroimaging, and pharmacological modulation of cognition. His work has been published in journals including Neurology, Journal of Neurology, Neurosurgery, and Psychiatry, Proceedings of the National Academy of Sciences of the U.S., and Lancet. These webinars are made possible through generous donor support, including a grant from Local 25 Boston Teamsters. If you'd like to contribute, please visit our website at autism.com. And now I will turn this over to Dr. Beversdorf.
Greetings, everybody. One good thing of good fortune I had forgotten at noon on Wednesdays is when we test our tornado sirens out here in the Midwest. Fortunately, my office is sufficiently secluded that I can barely hear it, which is great for this talk, but will be interesting when tornado season gets rolling. But nonetheless, with that a quick aside out, I'd like to talk a little bit about some research we've been doing on the role of stress or prenatal stress exposure in the etiology of autism. It's very interesting results we have found and put into context with the literature. Of course, I don't think I need to introduce what autism is to this audience, so I won't spend much time on this slide, but I will discuss that heritability estimates used to think that it was almost entirely genetic, but more recent work has suggested that with larger twin studies that there is some environmental component. Initial studies suggested that it was a fairly substantial component, but the more recent conservative estimate of a heritability of 0.83 seems to pan out. However, one does wonder as to whether when you look at both genetics and environment together, you might get something more interesting. Autism environmental factors have included parental age, advanced parental age on both sides, mothers and fathers, the gap between pregnancies being too short, too long. It seems to be a very modest risk factor. Maternal antibodies is one of the more powerful risk factors, which is not an environmental exposure externally, but is certainly a non-genetic environmental factor affecting development. Pollutant exposure has been reliably reported, again, a modest effect size, a modest relative risk, but nonetheless it has been repeatedly shown to be a factor. And early pregnancy lack of folate has been shown as well in a number of studies. One recent study suggested that too much might be a risk factor. However, it is an open question as to whether that group taking too much folate was taking it because they had other risk factors they were trying to compensate for. So that is an open question as whether that's reliable, but nonetheless it does deliver the message that there may well be a fine balance if it does turn out to be an issue. But gene by environment is very important. Some of these environmental risk factors have modest relative risk. It goes from a typical risk to a modest increased risk. But if you look at genetic susceptibility, we may be able to find out who really are those folks that are at greater risk of that environmental exposure and have a much better targeted understanding of what's going on. And prenatal stress is one of those areas that we have been exploring. A little over a decade ago, we published a paper looking at the stress exposures in mothers of children with autism and the timing during pregnancy. And as you can see, there was an increased history of these in mid to late pregnancy that was not present in mothers of unaffected children or those with Down syndrome as sort of another neurodevelopmental issue in case people were selectively remembering autism pregnancies because all the other stuff that's going on with them. That it was not observed with Down syndrome pregnancies at all. And also the timing of it would kind of suggest against just a recall bias, a specific spike at the end of the second, beginning of third trimester. A couple of years later, a group looking at Louisiana hurricanes found a very similar result, that the prevalence of autism was increased if there was a tropical storm or hurricane in the tracked Louisiana areas in the 5th to 6 months of gestation, and the severity of the storm was for their particular locale seemed to impact this risk. And a number of other studies since then have supported an association between prenatal stress and autism in large epidemiological studies. There was one that was a negative study, but if you looked at maternal psychiatric comorbidity and left that factor in, it seemed to actually still be positive. And the latest data is a group in the Southern Hemisphere that looked at that showed that suggested that the people with prenatal stress exposure have a bit more severity of autism, suggesting that it is a little bit of a different subtype, which may be very important down the pike. We were very interested in serotonin system. And the reason is that serotonin is associated, it's increased blood serotonin is associated with autism. There is also altered serotonin processing in the brain to some extent in autism. Serotonin is very important in neural development, but of particular interest there in the promoter region of the serotonin transporter protein. There's a variation, so there's a short version of the promoter region and a long version of the promoter region that affects serotonergic function, and this has been well known to affect stress reactivity. In most studies, the presence of the short version that lacks a few of the base pairs in this promoter region is the one that's associated with greater risk of depression after life stressors, for example, and greater reactivity of the stress response area of the brain and the amygdala to emotional stimuli. But it's also been inconsistently associated with autism itself, although there are as many negative studies as positive studies, and that led us to ask the question, is this because this is a stress reactivity factor maternally, and that it requires the presence of stress, and that's why the data for the gene itself has not panned out. We actually began to look at this experimentally. This is led by my former grad student, who is now a postdoc at the Mind Institute, Karen Jones, and we did a mouse model study where we looked at prenatal stress exposure, and we had mice lacking one copy of the serotonin transporter protein, which led these mouse to have a very similar level of SERT function as people that have the short version of the promoter region. And in this way, we could control both the stress and the genetic maternal susceptibility independently. The stressors were developed by Tracy Bale, and there's a variation in the stressors. We do different things each day so they don't get used to it. 24-hour novel objects, overnight light, overnight sound, red fox urine, they smell a predator, wet bedding, 10-minute restraint, frequent cage changes. These are annoying, they're stressful, they increase cortisol, but do not affect diet. So they're still physically healthy aside from getting enough stress to develop cortisol. And we then looked at their sociability, and Jackie Crawley has developed this sociability assay whereby you see whether the mouse here tends to approach another mouse or an empty cup, and also when you have a new mouse added to the empty cup, whether they spend more time with the new mouse versus the old mouse. And what we found with this study was that the mice that had no genetic alteration and no stress exposure had a preference towards approaching the new mouse, approaching the mouse. And if the maternal genetic alterations present, that was still the case. If they're exposed to stress alone, that was still the case. But they lost that preference for for interacting with the new mouse if they were stressed and there was a serotonin, in the maternal serotonin mutation as well. That's fine and dandy to show that in a mouse model. It's proof of concept that there might be something interesting, but that's an open question as to whether this has any relevance for humans. Is this relevant for autism? And we initially did a small study led by Pat Hecht, who you see up there. He went on to do a postdoctoral fellowship in Southern California, and he looked at mothers of children with autism, and we did the genotype, and we repeated our stress survey with confirmed diagnoses at our medical center. What we found was very interesting. The mothers that had the S allele, 1 copy or 2 copies of this serotonin transporter protein, had this in the 5th and 6th and 7th month of gestation, a history of prenatal stress. And if the mothers, the L allele that did not have any of the stress susceptible alleles, there was no association. So if the study that we showed you earlier, the studies that show that stress is associated with autism, prenatal stress exposure, if that's being mediated by maternal stress susceptibility, this is exactly the pattern that one might expect to see. And that was true for both the number of stressors and the severity of the stressors, whereby we took the scores of the severity of the stressors based on pre-existing data and looked at how much and weighted the stressors accordingly, so that more stressors would be weighted more heavily, and more severe stressors would be weighted more heavily. Now that's a very small sample, so it may not be completely convincing. So we happen to have another group that was looking at this at the same time, Zedong Liu at Queen's University, and what we did was we actually published this together. We said we'd look at their data and see what the results were, and they had a slightly larger sample, and we found exactly the same thing. The ones with the short allele at the 5th to 6th month of gestation had a higher incidence of prenatal stress only for the ones with the short allele, not for the ones with the long allele. And they also had enough data, we collected this as well, but didn't have enough siblings. They also looked at the typical siblings of the same mothers, and what they found was nothing at all. And that's really, really important, because if this was just the genetic marker was causing mothers to remember stressors, then one would expect the same pattern here. But this seems to be specific relative to autism. So looking into this, what might the mechanisms be, and what might be important in ways of mitigating this effect? So we'll start with the latter question first. One of the things that we actually wondered initially was, is our diet contributing to this? We've all read the reports of an increased risk of autism over time. There's some debate about whether obviously this is detection and not really an increase in risk. But nonetheless, we thought we'd look and see if our change in diet, we have a very significant change in our omega-6 to omega-3 fatty acid ratio. Omega-3 is the anti-inflammatory fish oils that you may hear of, and omega-6 is the opposite, actually, although it's not quite so simple. There are some omega-3s that are less impactful than others, and some omega-6s that are less problematic, and the data is not 100% in one way or the other. But we thought we'd look at it because also children with ASD have a high omega-6 to omega-3 ratio. Of course, that could be their diets. There's also change in American diets in that the omega-6 to omega-3 ratio has been increasing, such that the influence of omega-3 is less in our diets. And also this is important in stress reactivity and the inflammatory responses as well. So and this also demonstrates the evolution of men to include the McDonald's diet. And if you have the earlier hunter-gatherer diet, was 2-to-1 to 3-to-1 omega-6 to omega-3. Now it's 15 to 17 to 1. And in fact, if you have this guy's diet with carrying the can from the fast food joint, it may be as high as 50 to 1. The Coke not being the issue, of course, but the fast food diet. So Karen Jones actually assessed this by looking at a relatively omega-3 deficient diet, and basically our question was, would this supplant the maternal stress susceptibility genetically? Would this actually be sufficient to induce, in combination with prenatal stress exposure, to induce autism-associated behaviors in the offspring? And what we found out is that it didn't matter whether or not there was a stress. Those with a high omega-6 to omega-3 ratio diet no longer had a preference to spend time with the stranger mouse, and this was also the effect if you looked at just the area around the cup. It's not just when they step into the chamber, but they actually spend time close to the mouse. The normal diet, the offspring did prefer to spend time around the mouse. However, they no longer had that preference with the high omega-6 diet, low omega-3 diet. Well, we wanted to look then and see if this could be something that could mitigate the prenatal stress effect, and look at multiple diets and omega-3 deficient and omega-3 rich. The DHA is a specific type of omega-3. It's the most bioavailable one, and the regular, the AIN93, is a regular diet. So we basically took both studies and combined the heck out of them, creating a very, very complex design. We did replicate our finding with the regular diet that the stress exposure in mothers that had lacked one copy of the gene lost the social preference. And this very, very, very complicated design here, basically the punchline is that with the regular diet, the stress group with the maternal stress susceptibility did not show a social preference, although they seem to regain it a little bit with the DHA-rich diet. Now, whether these two are significantly different or not is challenging, but it gave us a hint that there might be something there. So we looked at it a little more extensively. And I don't have Fumihiro's picture, but he was my postdoctoral fellow. He is now on faculty in Japan at Kyoto Prefecture University. We took DHA and started before gestation and carried it all the way through to really just maximizing any potential rescue, allowing it to build into the mother's system prior to even becoming pregnant, and so that during the pregnancy they have heavy DHA exposure, and all the way through we continue to give the DHA-rich diet to the offspring to maximize the impact. And with the prenatal stress and the stress exposure and the SERT knockout, the maximized stress exposure, we found very interestingly an increase in dopamine, which is a chemical messenger that's not typically talked about with autism, but for this particular setting it may well be important. And again, this speaks to the importance of understanding that there are many different subtypes of autism. And these individuals, there is a little bit of dopamine in the area called the striatum, where there is already a high concentration of dopamine, but the prenatal stress exposed ones that were genetically susceptible was markedly increased. And of course they had excessive self-grooming behaviors as well, and some modest change in the 3-chamber test. It wasn't as robust as the previous studies, but nonetheless a general trend was observed. However, if you gave DHA all the way through, these individuals, the dopamine changes went way down. It went all the way back down to what the normal mouse had. And also the self-grooming went way down as well. So this is really intriguing. It's not something you can completely hang your hat on because it's only a mouse model, but nonetheless it does raise the question as to whether the effects of prenatal stress can be mitigated by a relatively safe anti-inflammatory dietary change, something we'd love to be able to look at a large epidemiological population to see if that in fact occurs with, say, natural disasters or something. And the 3-chamber social approach tests also, they recovered their approach behavior. sniffing the other mouse with the DHA diet, whereas the prenatal stress exposed ones, which are the control group without the diet, did not. The effects of DHA on anti-inflammatory and oxidative pathways. I've had the great fortune of meeting Grace Sun, who is an emeritus professor here at the University of Missouri, who is a biochemist, and she has written extensively on DHA and a bunch of other agents. She actually had multiple large Alzheimer's grants looking at this, and she the wonderful experience of her becoming excited about what our lab was doing with DHA and neurodevelopment. So what she's done with us is she's looked at byproducts of DHA, HHE, and HNE, and HHE is a byproduct of DHA that suppresses inflammation, like nitrous oxide, and HNE seems to increase it. If you give DHA, it increases the anti-inflammatory HHE, and if you give lipopolysaccharide, you create inflammation, which increases HNE. And if you give DHA, it recovers that inflammation, inflammatory HNE. So we looked at how this would affect the brain and plasma, and if you give DHA in plasma and in cortex, it increases, and HNE, it decreases, and the ratios are significantly better with DHA being given. So we're beginning to, and we're also looking at that, we're now looking at this in the prenatal stress model to see if, in fact, this is affecting offspring development, and seeing if we're beginning to now uncover the biochemical mechanism by which this acts. Also, epigenetics is becoming very important. We're getting into the weeds scientifically here. Epigenetics are modifications to the genes or gene products that can seem to be inherited or passed on. It's actually been a little more loosely used to how expression of what otherwise would seem to be genetic is altered. I'm speaking fairly loosely there. We'll give a couple examples, but it turns out epigenetic mechanisms are very, very important in the effects of stress. So we kind of need to look at that in our setting. And what we've done was we took our model, in the same group, Zedong Liu's group with Calvin Jarda in Queen's University, it happens to run a very large epigenetics program up there, and was able to run the analysis on the very same mice that we looked at earlier. We'd saved the brains of these mice and looked at gene expression of in these mice. So we looked, if you look at environmental conditions such as stress and maternal factors such as genotype, we were able to look at gene expression, the transcription. We look at the DNA methylation and microRNA expression. What DNA methylation is, you get a methyl group attached to different pieces of the DNA, and it generally suppresses the transcription of that DNA. And microRNA are small little pieces of microRNA that have powerful regulatory effects that we're only beginning to understand. And all of these seem to be affected by stress exposure, and in particular prenatal stress. So one thing we discovered is that with differentially methylated groups, that there was the hypermethylated and hypomethylated changes occurred. If you looked at the SERT and wild type mice versus controls, there were significant alterations, and if you looked at the number of methylated areas, that those with both the SERT and stress exposure were significantly increased. Now remember that by methylating, you are decreasing expression. So this increase actually is suppressing the expression of the genes. So we looked at it further and looked at effects on transcription. We found a number of genes that were affected by stress, affected by the SERT gene, and the overlap between those was fairly modest. But if you look at the upregulated and downregulated genes, when you looked at stress and effects, there was typically a similar number of the increase and decrease with exposure to stress. But if you looked at the SERT stress group, in the genetically altered ones, there's a much smaller number of increased expressed genes in this group, suggesting that if you have the serotonin transporter protein maternally, there's less ability to have this increased gene expression in response to the stress exposure. And finally, we looked also at microRNA expression and found the same pattern. If you look at wild type mice and compare them to the ones that are exposed to stress, there's a lot of increased microRNA production. If you look at the ones with the maternal mutation, there is a significantly decreased number. It's much less ability to generate this response to stress. So what this makes us wonder is if these gene expression and microRNA responses to stress are actually adaptive and helping the organism offset the effects of stress, but with the maternal alterations, they can't do that. And if you looked at what was being affected by these, this is another thing showing similar alterations, what's increased and decreased. But if you look at the targets, neurotransmitter transport and synaptic transmission and cell signaling are the things that are affected quite significantly, the differentially expressed genes. So that's all fine and dandy that we have something interesting going on in the animal model. What's this have to do with the clinical model? And it's the nice thing about what we've been able to do here is go back and forth between a translational model and a clinical model. Well, Zora Talabazita at University of Missouri, Kansas City is our genetics collaborator for this. We obtained an internal an intercampus research fund grant to begin to explore this. And what we did was we took the exact same blood samples that we looked at and described earlier when we looked at maternal genetics and stress exposure in the clinical population of mothers of children with affected children with autism. And we found if you look at the differential expression, so which genes are expressing RNAs and also differentially expressed microRNA, we showed a distinct pattern of the stress versus no stress group. 77% were upregulated. 2 of these were actually from the exact same group of microRNA that were found in the previous study in the mouse, which is really quite interesting. And another one has already been associated with prenatal stress. So there's an overlap between what's happening in the blood of mothers of children with autism and what's found in the mouse model in the brain. We need to explore that further in order to understand what these are doing and why they're important and what can be done about them, obviously. There's also 20 microRNAs that were differentially expressed by maternal genotype. 5 of them were also among the stress-dependent microRNAs from the previous study as well. And if you look at these individual groups, these are those genes that are significantly affected by stress and maternal genotype in this clinical sample. And so this is a little out there in the woods, but it's something we're trying to figure out what's going on. Why are these genes affecting it, both from a biochemical and from an epigenetic pathway? And a couple of other interesting highlights. These microRNA are also found the top hit for these microRNA expression profiles is dopamine regulation, which ties right back into what was increased in the mouse model and what we will begin to look at to see if we can reverse with omega-3 fatty acids, specifically DHA. And furthermore, It is remarkable because these blood samples are taken from mothers who had their child 6 years earlier, and yet we're still seeing long-lasting epigenetic changes. This actually has been reported in other settings. In fact, smoking after cessation has been found to create very, very long-lasting epigenetic changes as well. There's one other piece to this story that is of significant interest, and that is maternal antibodies to fetal brain. Our colleague Judy Vandewater, who also is working with my former graduate student Karen Jones, she is reported on, actually a number of you may have met her presenting at previous conferences. She has reported on maternal antibodies to fetal brain proteins. These are the band patterns that were shown in the past, but she has also specifically identified these antibodies now and what their brain targets are. But given the interrelationship between stress and immunity, we are very interested in moving forward to look at this, and we're now beginning to explore whether they're related. I don't have the results of that just yet, but we do know that these antibodies aren't just innocent bystanders, because in an animal model, they induced human antibody to fetal brain from mothers of children with autism was found to induce rather remarkable stereotype behaviors in the offspring in animal models, as has previously been shown. So this is not just a theoretical innocent bystander that's found as a marker in autism. It does seem to have some etiological role in repetitive behaviors, at least in animal models. So this work is not done in isolation, not even close. I'd like to thank a number of individuals. Basically, the Research Council grant with the biochemical data, Grace Sun and Mike Greenleaf. Kevin Fritchie is my omega-3 expert and colleague, and then students and research assistants that helped out with that in Dr. Sun's lab and Dr. Greenleaf's lab. Our epigenetics team here at the University of Missouri and UMKC is Dr. Rivera and Dr. Talbazita. And Dr. Hagen also helped actually gave us some of the mice to get started. Dr. Will is our collaborator for the animal behavior aspects. Brad Ferguson and Brianna Killey helped out, and now we have Taeson Wu helping out, and Judy Vandewater and Karen Jones moving forward. And with the previous clinical study, I showed you the work of Taeson Wu was a new student, but Fumihiro, Karen, and Pat. The same team was involved with the early studies. Mike Tilley did our early genotyping, and the Canadian team included Susan Connors, who actually connected us and was the reason why this study was able to be done together, and Zhudong Liu and his team, Calvin Sharda and Melissa Hudson. There's our team from a couple years ago. There's Fumihiro. It's the only picture I have of him, so I thought I'd highlight him. And Karen Jones had already moved off that picture as Pat Hecht. There's Brad Ferguson. I'm hidden in the back, which is probably a good thing. And there's Brianna, and these were the two that did were involved in our propranolol and brain imaging studies, a couple of papers of which have recently come out. And of course, this is never done in isolation. Unlike our other studies, our trial studies that were done with fairly significant larger funding, this is actually something we're just now getting off the ground. Internal Mizzou Advantage funds, Kansas City Area Life Sciences Foundation, the MU Mission Enhancement Fund, and the IDIC grant were what got us started down the pike with this work. So with that, I will close out and open to questions.
All right. Oh yeah, just leave your title slide up. That's perfect. I forgot to mention that beforehand. If you always leave that open, that'd be great. So we have a lot of questions. One of the first questions, and probably one of the easiest ones to answer for you, would be what kind of dosage was used for DHA during the study, or did it vary?
It varied. Let me pull it out as 1 milligram per kilogram, so it was mouse model stuff. And also bioavailability is very, very different from mouse to human. So I wouldn't take that to the bank just yet. We need to be able to do more with that. that's what we use in the mouse study, but I wouldn't translate that yet.
Okay, and what kind of source were they using, the DHA source?
I would have to defer to my colleague Kevin Fritchie on exact source, but he went to a company that made it fresh. We kept it stored under very specific conditions, and we added it before adding it to the chow. So he went to it's listed in our paper the exact source,
Okay.
but he guided that actually.
Okay.
You need to have somebody that really knows this stuff because it does degrade significantly, and that's a very important issue for anybody looking to use these.
Got it. And so it would have been it was directly provided by a company, though. They weren't distilling or do anything like that. Okay. So
that's right.
They're using a manufacturer. Yep.
He also tested it to make sure that it was still viable.
Okay.
Beforehand.
Got it. Okay. One question we got really early. So what is too long between pregnancies?
I don't remember the data on that because it's not something I've explored. And again, it's just an epidemiological thing. There may have been other factors, but I'm sorry, it's not my area. But there was some data suggesting that the longest spans of pregnancy. Now, who knows why that may be important? It may be because there was stressors going on in the lives that delayed the pregnancy. This is epidemiological data, so I don't put too much weight on that as of yet. I think we need to confirm it with other studies. That's one of the epidemiological data can sometimes have entire countries as their data set, especially in Scandinavia where they have these wonderful national databases, but you don't know what else might be accounting for things. It may be something else going on that's driving this that you haven't been able to account for in your variables. So I don't know the exact number there. The literature is out there, but it's not exactly my area, but I don't hold too much weight on it till we know why.
Got it. And I've read that too. I think 7 years was the interval that I heard in a couple of studies, but it's definitely something that they can look up. If they Google it, it should come right up.
Sorry, I don't know the exact number. 7 years sounds right because that sounds like a longer term.
Yep.
But it hasn't been my area, so.
Okay. You talked a little early on about folate, and I know there are different kinds of folate. So we had questions about what types of folate are implicated or what types of folate might be relevant in these discussions. Do you have insight about that?
The early studies were just looking at folate, but that's a good point because there are a lot of variants of it that have different bioavailability, and people who have other genetic issues may have difficulty with utilizing those if they don't use the proper form of folate for their individual needs. That's a very important issue that's been looked at by a number of other investigators, many of whom are actually well associated with the Autism Research Institute, and they'd be the ones to best speak to that because they have their feet on the ground. I've just simply looked at their papers and noted that we may need to go beyond just looking at folate by itself and look at specific types of folates that have more bioavailability for those people that have greater need or genetic susceptibility that causes them to need this specific type.
Right. It sounds like a good future webinar.
Right.
Right. And we do have some other talks on our site that touch on this. I think one of the genes that comes up is MTHFR. And I know that some of our other speakers have talked about that in their webinars. So if whoever asks that question, if they want to go look at our library, they'll probably find some good resources on that. Okay. So another question that we got was about, they were asking about short intervals in pregnancy. I know you already talked a little bit about trying to find that information elsewhere. But what about this? This person is asking a specific question about continued elevations of IL-6 have been noted by different groups as posing a risk for untoward outcomes. Many different supplements have been posited as agents to address this issue. Do you have any specific thoughts on this, or are you familiar with that research?
Yeah, I am a little bit. because that's another factor that could well be related to IL-6 is also a stress reactive cytokine, and I don't know the relationship between that and IL-6 as of yet. Is IL-6 part of what's going on here? That's, I think, a very good question, and it may well be that whatever we target here will target that as well, and maybe they have overlapping mechanisms, which is actually one of the reasons we're looking at the maternal antibody factors. One of the problems is when you're trying to get funding for grants is looking too disjointed, and we're trying to get some pilot data to show the linkage with inflammation, because you have to prove that there is some association before they'll give you money to look at it further. So we haven't been able to branch into that area, but it's an excellent question, and I suspect the answer is yes, that this is related. IL-6 may be part of this. It may be part of the mediation of it, frankly, and whatever targets one may target the other. So DHA would at least be on the playlist of what you might want to look at there.
Okay, and continuing on that, this person actually said how much of this information can be translated into prophylactic measures for women who are preconception, especially based on recent findings by Fair at OHSU and Littman at NYU regarding any level of inflammation offering potential input to adverse outcomes via IL-6.
Yeah, Damon Fair's data is really interesting. I want to answer that question so bad, and try to find a way to do this. I mean, obviously, one thing I'd be curious of is, does DHA, is that going to be an important factor? And I've been starting to talk to some folks that do DHA work to see if we can sort this kind of thing out. I wonder the same thing. I'm wondering if these are all interrelated factors that are affecting development. And of course, from that early slide I showed you, that the developmental outcomes in these affected individuals that are prenatal stress associated may be more severe, and that would fit the Damon Fair's data as well. Damon's I was excited to see that paper come out. I think, and I think we all may be looking in the same direction. I don't know the answer. I'd love to know the answer. I agree that it's a great question, and I want to answer that.
Okay, great. Yeah, I didn't mean to put you on the spot. It's just it was an interesting question.
Where I want to go. It's all good.
Yep. So here's a change in subject. They're asking about stressors considered to be significant, causing stress during the critical period in pregnancy. So you gave great examples that were used in studies of, I mean, tropical storms and hurricanes are great because everybody's experiencing the same thing. Are there particular stressors that are suspicious? Are there things that people are thinking might be relevant universally?
We don't have an answer for that just yet, but in our study, it was psychosocial stressors, and it was quite a gamut of them. Death of a family member, a loss of a job, a significant move, and we took the old Holmes and Ray scale, basically, real simple. That one where 100 points is death of a spouse. Nobody in our study had that, but nonetheless, that's the scale. And 71 is divorce, and so and basically anything that scored above 10 we counted. And so, and in aggregate, that big list of psychosocial stressors did reveal this. So basically, we don't have any evidence to suspect that there's one type or another type. It's a really good question. It may be very important going down the pike, but in a study where all these were kind of aggregated, that seems to be important as a group. So there didn't really seem to be something that pointed us in one direction or another. Now, the stressors that have been published elsewhere, bereavement stress, death of a family member was done in one of the Scandinavian studies, obviously the hurricane study. So those are a couple examples that one can point to specifically, but there's nothing to be certain that those are the only ones. So severity of psychosocial stress seems to be important from our data and also from the hurricane data, the severity of the storms at least, it would be fantastic to have a biomarker to begin to address this, and we're hoping to get a chance to look at that in the near future.
Okay. Here's another tough one. See what you think of this one. He says, do you put any clinical validity into urine sampling for serotonin and dopamine levels? Literature continually debunks this, but many parents are courted by practitioners recommending this type of assay in order to address a variety of issues. Do you have any thoughts about that, or have you seen that?
Well, so the serotonin in the urine, you're processing it. The cerebrospinal fluid was actually the thing that's looked at the most closely and carried the most weight. Of course, that's not easy yet. And there were some abnormalities found in the cerebrospinal fluid. And then somebody pointed out that the stress of getting the spinal tap might be very important in that stress reactivity. I don't know about urine studies, but I can say a couple things. Number one, dopamine is not universally altered in autism. It may be very important for certain subtypes, and that's kind of what we're thinking is going on here. Serotonin is elevated in a certain percentage in blood. It's platelet serotonin. It's elevated in a certain percentage of blood of patients with autism, but not all of them. And the significance of that is not completely understood. So I'm not sure what urine markers would add to that picture beyond what is already known from the blood, and the fact that dopamine may not have any particular role for autism as a broader group. Although it's interesting because with our changes, maybe dopaminergic targeted therapies would have a different effect in prenatal stress-exposed autism as opposed to ones without. It's an interesting question, but one that's still hanging out there. But again, I'm not sure. Urine seems a little bit far from the source, if you will, for what we're looking at in the brain and in other systems. Although serotonin is also very important in the gut, but that, of course, isn't the urine system either.
Okay, great. This is a question about cortisol levels. You touched on this just a little bit. Are there studies that address specifically concerns about cortisol levels in ASD, either upgraded or downgraded, or?
Actually, the cortisol data on autism is interesting. We just published a study looking at cortisol reactivity in autism. It's a bit all over the map, but we've actually shown that those with greater cortisol reactivity tend to have greater gastrointestinal disturbances. So I think this is all going to tie into subtyping. Cortisol reactivity in autism may be important, but particularly important in a subtype of autism, those with a greater anxiety comorbidity in all likelihood, and anxiety is a very significant comorbidity in autism. So I think cortisol per se is going to become more important as we understand the subtypes of autism.
Okay, and here's another question from that same person. They're asking, considering vitamin D and DHA involvement in serotonin metabolism, have there been tests of the synergistic effects on maternal stress?
I don't know of any synergistic work, but vitamin D is very interesting, and it seems to, we're just now beginning to understand its role in development. And something very interesting, in my clinical population, I keep finding individuals with autism with low vitamin D levels, and again and again and again and again. There's mixed data in the literature on that. Maybe it's something about Missouri kids, I don't know, but it's almost ubiquitous that they're somewhat low. It's quite remarkable. Maybe it's lack of sun exposure. They're not out playing with the other kids so much. I think it's a really, really interesting question. It could be very important, but we don't know yet.
This is about different points in the pregnancy. So is stress more significantly impactful at different trimesters?
That's the funny thing, is our data and the hurricane data suggested mid to late. There was a study in the Nurses' Health Study that showed that early in pregnancy, a history of abuse was more important. Now, maybe the timeline of that wasn't as specific, so maybe that carried over into the middle of the pregnancy. It's hard to say, but there's a couple of lines of evidence that suggest later. That one suggests earlier. There's a third study that suggested third trimester as well. I can't remember what the stressor, I think it was one of the bereavement stressor studies. So it's a bit of a mixed bag, but tends to suggest later pregnancy stress. But there is actually one other study, the Nurses' Health Study, Mark Weiskopf's group, also showed that early life, major stressors and history of abuse actually can increase the risk of subsequent offspring having autism, suggesting something really important about that epigenetic mechanism that we're talking about. So that's really intriguing. So that kind of throws you off the entire pregnancy scale into lasting epigenetic changes. So it seems to be later pregnancy that's important. The mouse model goes with this, but that's not 100% universal across studies.
This next question, they're bringing up Dr. Michael Cooperstock, who I believe is at University of Missouri, and
Yes.
asking about connections between infectious conditions and immune dysregulation like PANS and PANDAS and serotonin issues that you've touched on. So have there been any connections there that have been identified?
I actually know Mike Cooperstock. We've had several chats. But I don't know how it relates to the serotonergic system. He might know better the neurochemistry of the specific PANDAS syndrome. But it does speak to the, I don't know how stress relates to this, but it clearly speaks to the importance of immunity in these conditions, in neurological, neuropsychiatric conditions. These are a really interesting group of patients that have these really bizarre neuropsychiatric conditions that develop later in life, not early like in autism, but later in life, and they come out of the blue and are associated with this inflammatory infectious response. We're kind of, thanks to Michael, we're beginning to understand a little more broadly the spectrum of this. We used to think it was a very specific syndrome, and it looks like it may be broader than that. And then immune therapies target this. However, it is fairly distinct from autism spectrum disorders because of the lateness of onset. These individuals are doing fine until this happens, but it does speak to the importance of the immune system in neuropsychiatry.
Okay. Well, I think we have time for one more question. I know we're almost out of time. It's gone really quick. But this is a question about supplementing with DHA and EPA. They're asking about whether or not this person's saying that they've seen increases in stimming and hyperactivity with those supplements, and if that was observed in the study with the mice, if there was anything like that.
We actually had a decrease in repetitive behaviors, but let me give a little quick caveat. This they did not show the benefit was not reliable if we started it after weaning, which is the time frame when you would start for treatment of somebody with autism. And again, the other caveat is that this is targeting just prenatal stress associated. So we're giving it through pregnancy, and also we're giving it targeting this one population. The data for DHA in autism in general is mixed at best. One large study showed no benefit. And so prenatal giving the medication is different than giving it after diagnosis. And also we're looking at a specific subtype. So I don't know how, if at all, this effect generalizes to the broader autism population. So increased stimming may well occur with administration of DHA to the general autism population, but that's kind of a different thing than what we are looking at. In fact, it's a bit of a challenge because we have to point out, beat over the head grant reviewers, hey, no, this is, yeah, we know that that trial was negative. We're giving prenatal medication. Hello, that's different. So hopefully we can get that point across in order to move forward with this on a larger scale, because this is actually not a trivial group. It's 10 to 15% of autism, at least according to the studies that we did, the ones that had the stress susceptibility and stress exposure from our entire block of autism families was 10 to 15%. It's not a trivial number, and if you could actually improve the life for these individuals, mitigate the more severe effects of it, that would be a big save. So I would argue it's an important thing. We're not there yet. We don't have an answer, but I think it's an important avenue to pursue.